Okay, so this is chapter 22, Fungi, Evolution, and Diversity. In terms of the outline of the chapter, we'll talk a little bit about fungi evolution in general and characteristics of fungi in general, right? And then we'll talk about different types of fungi that exist. And then in particular, talk about symbiotic relationships of fungi because there are a lot of fungi that form these symbiotic relationships that then impact the ecosystem or obviously impact another organism. Okay, so there's about 100,000 different species of fungi. I think when most of us think of fungi, we probably think of mushrooms, right? But there are many different types of fungi. Mostly they're multicellular, and they share a common mode of nutrition. So heterotrophic, and a lot of them are also saprotrophs. And again, we've mentioned this term before. Make sure we understand what that means. Again, the organism or the cells will release digestive enzymes that will then break down molecules, right? And then they can absorb those more simple molecules. So in terms of evolution, about 1.5 billion years ago, so these guys have been around a long time. And so it says plants, animals, and fungi trace their ancestry to protists. So again, remember when we were talking about protists in the last chapter, we had those 6 different supergroups, right? And we said that protists that are members of the supergroup Epistoconta are more animal-like and fungi-like. So fungi are not really are more closely related to animals than they are to plants. Again, remember, they're within this supergroup that's shared with animals, okay? So that's what it's saying here. All right, so this is a flagellated unicellular protist, was most likely a common ancestor of fungi and animals, and we kind of talked about that in the last chapter. One thing about fungi is that they don't easily fossilize, so it's hard for us to determine the sort of evolution and the history of them because there aren't many fossils. Okay, so it said these guys probably evolved a lot earlier than that earliest known fossil, which is about 460 million years ago. Okay, so again, most likely they were around longer. They just don't fossilize well. Mycorrhizae is an example of a symbiotic relationship of fungi with plant roots. And you can actually, in plant fossils, you see these mycorrhizae around. So suggesting, obviously, that they were around, at the time, and around a long time in terms of helping plants out, in terms of them absorbing nutrients from the ground. Okay, right? And so it's basically kind of, just like I mentioned here, that if these fungi weren't around and didn't associate with those plant roots, the plants may not have as easily diversified and flourished. Okay, so the fungi probably played a pretty important role in allowing plants to basically colonize land. All right, so just like we looked at before, this is the same thing, right? We're going to kind of zoom in down here now. Okay, so again, 6 supergroups we talked about when we were describing the protists. Now we're in epistoconts, and you can see fungi here. So we're going to particularly talk about different types of fungi. Okay. So again, if we zoom in now on fungi, these are these 6 groups within the kingdom fungi. Okay, so it says AM fungi were once considered part of Zygomycota. So anyway, these are kind of 2 separate groups. At one point, they were one group together. So again, this is true of taxonomy, and I keep saying this throughout the semester that these groups change, right? Taxonomy changes based on more and more data that we obtain, okay? And so anyway, we're going with this. So common name and scientific name. So Basidiomycota are club fungi, right? Ascomycota, sac fungi. Glomeromycota, AM fungi. Zygomycota, zygospore fungi. Chytridiomycota, zoospore fungi. And then Microsporidia are obligate parasitic fungi. And so if you look here, too, in terms of the star, that these guys were recently placed within the kingdom fungi, okay? And we'll talk a little bit about them. And we actually didn't even talk about these when we did the fungi lab, or when we're going to do the fungi lab. All right, so again, here's where we're at, right? Here's fungi, and now we're talking about the different groups of fungi. Okay, so in terms of, again, fungi in general, sort of general biology of fungi. If you talk about the structure of the fungi and kind of what it's made up of, okay? So it says the body, right, or the thallus of most fungi is multicellular, okay? Our one, big example of a unicellular fungi is yeast. Okay? Yeast are single-celled. They're used a lot obviously in production of beer, wine, bread. They're also used a lot in the lab in terms of understanding processes that happen within a eukaryotic cell, okay? The body, though, what it's made up of, if you kind of think of like the tissue of the fungi, it's a network of thread-like hyphae. Again, think about those strawberries that we had in lab with the mold growing on them, right? Or strawberries that you've seen or stuff that's growing mold. It's kind of fuzzy and sort of thread-like, the sort of structure of the fungi. And so it's made up of something called hyphae. And you can have different types of hyphae. So septate hyphae have their hyphae with cross walls. I'll show you a picture in a second. Non-septate hyphae or non-septate are multinucleated, so they don't have the cross walls. So think of these guys, these hyphae, kind of as what makes up the tissue of the fungi, as different types of cells that make up the tissue, okay? It says, so the mycelium, right, is made up of these hyphae, and that gives a large surface area per unit volume. Cell walls, right, of the cells are made up of something called chitin. And chitin is a component of insect exoskeletons, okay? Again, showing this sort of relationship between fungi and animals, okay? Excess food is stored as glycogen, just like animals, okay? So we've kind of been talking about the sort of different types of carbohydrate storage molecules, like even when we were talking about in protists, right, that there was kind of some variations within there. Here, again, all animals will store glucose as glycogen. Plants will store glucose as starch. Fungi store glucose as glycogen, just like animals. All right, so this is showing you. This is the mycelia, right, essentially, and it's made up of these hyphae, okay? So again, this sort of tissue, the body of the fungi is called mycelium, and it's made up of hyphae, which essentially are the cells, right? And so you can have septate, right, with these walls. So here's a nuclei, right? Or you can have aseptate, meaning there's no cross walls and you just have this sort of multinucleated sort of, I guess, fibrous strand, okay? In terms of reproduction, and again, when we did this in lab, or when we're going to do this in lab, we talked a lot about these different types of spores and things like that. And so it's kind of used as one of the ways to sort of characterize some of these different types. But in terms of, in general, kind of same idea, right? So you can have both sexual and asexual reproduction, just like we've been talking about before. If you're talking about sexual reproduction, there's 3 stages, right? You'll have haploid hyphae. That's what this is showing you here. You have this dikaryotic stage where you have 2 nuclei, right? And then you end up with this diploid zygote, okay? Which then can go back to form more of these haploid hyphae and basically, continue that reproductive cycle. All right, so let's go through this a little bit more. That's kind of going back to that picture. It says hyphae from 2 different mating types will fuse, right? And you'll get this dikaryotic. That's where this comes from, okay? And then basically you could get these haploid, right? Eventually you can get these haploid spores that will form, okay? And then those spores will germinate and basically carry out development and make a new fungi, okay? In terms of the spores, and again, this is what I was talking about, kind of what we talked about in the lab, in terms of the different groups of fungi will have different names for their spores. But again, overall, it's kind of the same similar process, right? These spores are, a reproductive structure that, again, can be transmitted around, blown in the wind, what have you, and then eventually will form a new organism, right? A new fungi, okay? So obviously, for terrestrial fungi, it makes sense to have a non-motile, a wind-blown spore, right? So again, what's a spore, right? A reproductive cell that develops into a new organism without the need to fuse with another reproductive cell, okay? So again, once you have these spores, they kind of remain in a dormant state for a certain period of time. They end up getting dispersed around, and eventually, when conditions are favorable, they will form a new organism. Okay? In asexual reproduction, you can have these spores that, these windblown kind of spores that occur as well. Okay? If you're talking about sexual reproduction, you're kind of talking about these two different types of gametes that end up joining together. Okay, we'll talk a little bit, you'll see a little bit more about this as we go through the specific types of fungi. Okay, anyway, just to kind of put this in perspective, a large mushroom can produce billions of spores in a few days. Okay, so tons and tons of spores. And then when that mushroom breaks, those spores are released. Okay, if you're talking about unicellular fungi, yeast, they actually reproduce asexually by budding, right? There's no spores that are going to be released or anything like that. They just basically divide, reproduce by budding. Okay, so now let's talk about some of these different groups. And like I said, what we just talked about with the reproduction and the spores and stuff like that, it's going to we'll talk about it again as we talk about the specifics here. So this is just giving you a kind of little bit of history here. A scientist in 1969 basically kind of was the first to sort of categorize fungi as separate, right? So as being a separate group from protists, plants, animals, and photosynthetic organisms. Right? So it says fungi are the only type of multicellular organism to be saprotrophic. Okay? So again, suggesting that these guys are different. Okay? Again, we know this. They're both placed in the supergroup Piscicana, and here are the fungal groups that we're going to talk about. I already mentioned this. Okay? So like I said, they're differentiated according to their life cycle and the type of structure they use to produce spores, right? This is what I've been saying all along, and this is what we talked about in lab, all right? Again, it's similar in terms of their makeup and structure, similar, but you're going to have slight differences between the different groups. Okay. These microsporidians, like I said, were sort of recently moved into the kingdom fungi, and they're medically important because they cause disease. All right, so again, here's your table, groups, right? You don't see microsporidia on here, but these are the five main groups. We'll mention microsporidia in a second. In terms of reproduction, right, you can see the different names of the different spores, okay? And then key features and some examples, okay? Again, very helpful. Just like that protist table that we had, this is super helpful as well. I suggest, obviously you got to study, the entire chapter, but please, please study these tables, both from the protist and from this chapter. Okay, so for the microsporidians, we'll talk about them a little bit. Like I said, they sort of recently moved into this, and they're basically parasitic fungi that are going to cause disease, okay? And so they're obligate intracellular animal parasites. Obligate meaning this is what they have to do, and they have to get inside an animal cell to be able to survive, okay? So it says here that we used to think they actually were an ancient line of protists, but like I said, they've been recently moved into the fungi group. And so here's an example of one particular species, E. cuniculi, right? And other microsporidia, so there are other specific species as well, they can cause diseases, in particular in immunocompromised patients. So patients that have AIDS might end up with these types of fungal infections, particularly caused by these microsporidia. And most people with a normal functioning immune system won't develop these types of diseases because their immune system will be able to get rid of them, okay? So again, it says it infects hosts with spores, which will infect intestinal in particular, and also neuronal cells, and then basically will cause those cells, to die. I think I have a picture. Let me show you a picture here, okay? This is showing you a picture, right? So this is inside the cell, okay? And you can basically see all of these spores of the fungi inside the cell, okay? All right, now, chytrids are another type of fungi, aquatic fungi, okay? And so because they're aquatic fungi, it makes sense for them to have flagellated gametes and spores, right, called zoospores. Why does it make sense that they're flagellated? Because these spores are not windblown, right? They live in water, so they have to be able to swim around in the water, okay? So again, the spores that they produce are called zoospores, and then those zoospores will grow into new chytrids. So some will have an alternation of generations life cycle, which is super uncommon among fungi because remember, we said that alternation of generations is something common in plants. And, we now know that fungi are more closely related to animals. So you really don't see this much within the fungi. They play a role in decay and digestion of aquatic organisms. Again, remember they're aquatic plants. Some are parasites of living plants, animals, and produce, but a lot of them will decay, you know, dead types of animals in the water. Here's an example of a parasitic chytrid. Okay, it basically has decimated about 200 species of frogs. Again, they can survive on frogs, right? Because their skin is super moist. Their spores, they're flagellated. They're able to kind of swim along in that small amount of water. Okay, and so once it infects the frog's skin, it actually causes it to thicken. And because the frog relies on, you know, both getting oxygen through its lungs, but also a lot of its oxygen comes through its skin. That's why it's really necessary that frogs have this thin, moist skin. And so if that skin begins to thicken, they basically won't be able to intake oxygen, and they basically. So this is basically showing you here chytrid's parasitizing a protist, right? So this is showing you kind of the algae here, and then the chytrid, right, all around here. So again, they can parasitize. We gave an example of frogs, but also protists as well. Okay, so again, let's remember what group we were talking about, right? We were here. Where's our groups? Right? Chytridiomycota. So we talked about Microsporidia. We talked about Chytridiomycota, right? The chytrids, those aquatic fungi that form zoospores, okay? Microsporidia, remember, are parasites, basically they get into animal cells, and they're going to cause disease, mostly in immunocompromised patients. Now we're moving on to the next group here, Zygomycota, okay? Again, good idea to keep this handy as you're going through this. Sorry to jump back, but I want to make sure you guys understand where we are. So now we're in Zygomycota. So about 1,000 species, and they produce zygospores, hence the name Zygomycota, okay? Mainly they're saprotrophs. They decompose animal and plant remains. One example of a zygospore fungi, which we looked at in lab, black bread mold, okay? Scientific name, Rhizopus stolonifer, has both sexual and asexual phase of its life cycle. And if you look at the hyphae of the black bread mold, right, it has specialized types of hyphae. So again, remember, we talked about hyphae in general. That's what makes up the fungus, okay? When you talk about black bread mold, it particularly has specialized types of hyphae that have different names, right? Stolons, rhizoids, and sporangiophores. Okay, we're going to look at a picture and we're going to see those. And we saw the same exact picture that we're going to look at in a second in lab, also in your lab manual, okay? So if you look at the Rhizopus stolonifer life cycle, again, I think you should be familiar with this life cycle in terms of me. I could put the actual picture on the exam and ask you about it, but I don't expect you guys to memorize the stages or anything like that, okay? So I'm going to show you, I'm not going to go through these words. Let me just show you it from the picture and we'll kind of go through it, okay? But the one thing, again, remember, called zygospore fungi because, Zygomycota, right? They form zygospores, okay? The sporangiophores are what's going to house those spores. All right, so let's look here, right? So again, you can look at the black bread mold on the bread and look at those specialized types of hyphae, right? Rhizoids, okay? Where else are you? Sporangiophore, okay? You can see those here. And then the sporangium is going to house the spores, okay? And so again, you can see that hyphae of opposite mating types will touch, and that's what basically starts this reproductive cycle, okay? So again, be familiar with this, looking at it, be able to identify these different types, particular types of hyphae here. Okay, but for the most part, nobody needs to memorize all the, the steps or anything like that. All right. Okay, next group. So again, for Zygomycota, right, our example was black bread mold. Next group, Glomeromycota or AM fungi. Okay, so AM stands for this, arbuscular mycorrhizal fungi. Small group, about 160 species. Okay, so these arbuscules are basically these sort of branching, it says invaginations, or sort of branching, I guess, trying to think of another word. basically, I guess kind of, yeah, I mean, think of them as branches and sort of multiple sort of extensions that will invade the plant roots, right? And so again, it's going to keep making more because it increases the surface area. And so these mycorrhizae are a mutualistic association of the plants and the fungi. Both benefit from it, right? Mutualistic. Okay, so it says they were classified as zygospore fungi for many years, right? But like I said, they sort of separated out these groups because based on molecular data, they are separate. Okay? And we'll come back to talking about those a little bit more at the end of the chapter when we talk about symbiotic relationships, okay? But just remember, that's a whole separate group. All right. Another group here, right? We're in sac fungi. So again, if we go back, sorry to jump back, but let's put ourselves in perspective here where we're at. Right? Now we're here. Ascomycota, sac fungi. Okay? All right. So here we are. Lots of them, about 64,000 species of sac fungi. Okay? Again, most are saprotrophs. They will digest resistant materials that contain like cellulose, lignin, or collagen. And so therefore, they're particularly ecologically important, right? Because not many other organisms aren't able to break down those types of molecules. And so these fungi can do that. And then again, what they're broken down into is then available for other organisms. Okay? There's also, so one particular type of sac fungi called Neurospora has been used in some scientific studies. So just giving you an example there. Morels, truffles. Okay, we've heard of truffles before as like being expensive mushrooms. They're not mushrooms though, but they're a type of fungi, similar type of sac fungi. So there's kind of a lot of diversity here. This is what this is sort of listing here, right? So also they cause many plant diseases. So diseases called like powdery mildews, leaf curl fungi, ergo of rye, chestnut blight, Dutch elm disease. These are all just kind of examples of plant diseases caused by fungi. Also, they can cause human infections. So Aspergillus, right? We looked at that in lab. And Candida can cause human infections. Another type, Talaromyces, which basically is penicillium, is a source of penicillin. I'm not really sure why we renamed it, but anyway, it's sort of renamed. But we also looked at that in lab as well. Okay, so you can kind of see, and this is all within this one group, the sac fungi. Lots and lots of stuff going on within this group. Okay, if you look at the life cycle, okay, asexual reproduction. Again, we talked about yeast, our type of sac fungi. They reproduce by asexual budding. Okay, and then remember, yeast are single-celled. The other members of this group produce spores called conidia or conidiospores. Okay. All right. So, and we looked at this microscopically when we looked at Aspergillus and Penicillium in lab, right? In terms of what those conidia look like. Okay. The conidia of a certain species, certain genus of fungi causes allergy, so people can be allergic, right, to certain types of fungi. So this is basically showing an electron micrograph here, maybe not an electron micrograph, but high magnification of a yeast cell, right, and budding. Okay, this asexual reproduction here is showing you, right. This looks very similar. We looked at Aspergillus and Penicillium in lab. You can see this conidio, right, the conidia and the conidiospores with inside. Okay, so it almost looks like a stem and a flower on the stem. Okay, if we look at sexual reproduction, okay, so this was asexual reproduction, right? If we now focus in on sexual reproduction, you have something called an ascus that will form. And again, we saw this when we looked at the slide of Piziza, okay? And it's just the same picture that's in your lab manual, right? So this ascus forms, and within it are basically, spores, okay? So it says ascus may be surrounded and protected by a sterile hyphae within a fruiting body called an ascocarp. So the fruiting body basically protects these reproductive cells, right? And you can have different shapes. So in cup fungi, the ascocarps are cup-shaped. In morels, they're more kind of bell-shaped. Okay, So it says then these ascospores are released and dispersed by the wind. So let's look at a picture here, right? So this is showing you, so this is the ascocarp, essentially the fruiting body, right, of a cup fungus. This is showing you the fruiting body of a morel. Okay, again, type of sac fungi. And if you zoom in microscopically on this very edge here, right, again, kind of part of the fruiting body surrounds this, right? You can see the asci, they're multiple, and then you can see the ascospores within inside. And so if this breaks apart, all the ascospores are released, right? And then it can be dispersed. Okay, there are many mutualistic relationships between ascomycota and other plants and animals. So some examples, right? Leafcutter ants and some species of termites will keep fungal gardens within their colonies. Okay, and so they kind of help each other out, right? So the insects will feed and protect the fungus, and the fungus actually provides food for their developing larvae. There are also, like I mentioned before, right, kind of on the intro slide to these sac fungi, is there are, fungal parasites to plants, right, that can form diseases like Dutch elm, which can basically wipe out elm trees. Another type of sac fungi can actually cause a disease in bats called white nose syndrome. And so basically what happens is when the bats are hibernating, this fungus will kind of start to grow, the sac fungi, on their muzzles. And basically the infection will cause the hibernating bats to wake up, but they're supposed to be hibernating, there's no food available, and they end up starving to death. All right, so this is showing you these leafcutter ants and this sort of fungal garden that they keep. And then here's the poor little bats that are hibernating. You can see this white here. This is the fungus that's growing on them. Molds. Okay, and we kind of mentioned these guys as we've gone through. More sac fungi, aspergillus we mentioned, can actually cause disease. Some species. Others are used in the production of soy sauce, okay, because you can ferment soybeans using aspergillus. So these are helpful, right? So all of what's listed here, these again are all characterized as molds. These are sac fungi that are particularly helpful. So in the food industry, aspergillus can produce soy sauce, also used to produce citric and gallic acids. Yeast, obviously, make beer and wine, bread. Cheese industry will use penicillium to make blue cheese. I think that's a typo there, right? Blue cheese, right? Brie. Also helpful in other ways. So I said this before, I think, maybe I mentioned it in lab, but yeast in particular used in the lab a lot in terms of, again, understanding basic cellular processes in eukaryotes. Also, you have the ability to mutate genes and see how that affects different processes. Okay, and so that's there as well. So these are all ways in which molds or sac fungi are helpful to humans. Okay, they also can be detrimental or harmful, so they can cause or accelerate spoilage. And they can also release toxins, right? So I mentioned this before, that you can use aspergillus in a beneficial way to produce soy sauce and citric and gallic acid, but also if you have peanuts and other grains that get colonized by another species of aspergillus, it can actually produce a toxin, which can cause liver damage in animals. Okay, so you have to there's sort of all these sort of pluses and minuses to these. Also, another type of mold that's harmful, so candida, I think we mentioned this already, causes fungal infections. And so again, you see these types of fungal infections more in patients that have a compromised immune system. Okay, all right, so here's another species of aspergillus that can secrete a toxin that acts as a carcinogen. Okay, another type of these are all sac fungi that can produce another type of toxin. Okay, another one that causes athlete's foot. Another type that can cause something called the fungal flu. Okay, there are many different examples. All right, sorry, just to continue. All right, we're in another group. So there was a lot of diversity within the sac fungi group. Okay, a lot of things that are beneficial to humans. A lot of examples of species that are or groups of sac fungi that are detrimental or harmful to humans. So make sure you know these examples of of sac fungi that are harmful and beneficial, and can basically kind of answer that on an essay question if I ask you. Last group we're going to talk about is club fungi. Okay, so these guys are mushrooms fit within this group. You call them Basidiomycota, and there's about 31,000 species, so there's a lot. So like I said, mushrooms we're all familiar with. Other types of fungi that fit within this, so stinkhorns, shelf fungi, jelly fungi, bird's nest fungi. There are poisonous mushrooms. So there are about 75 species, I guess, that have been identified as being poisonous. One example is this death cap mushroom, and in your book there's a little bit more information on the death cap mushroom. And they also can cause plant diseases like smuts and rust. Okay. All right, so this is actually showing you an example or a picture of this death cap mushroom. Okay, so don't be going to eat mushrooms that you find out in the wild, because you never know. And this is showing you a picture of this ergo infection of rye, which is caused by a particular fungus here. See this guy here growing on there. All right, and so here are some of these, pictures of these examples that we just mentioned. This is a stinkhorn, bird's nest fungi, right? A puffball, shell fungus, and a type of mushroom here. Cool looking, right? In terms of reproduction, usually sexually, okay? So you have these opposite mating types that fuse. This is similar to what we talked about with the black bread mold. You'll form these basidiocarps. Again, Basidiomycota, named by the types of reproductive structures, right? Basidiocarps, which is what you recognize as a mushroom, okay? And then within them, they have something called basidium, okay? And basically, you'll get the formation of basidiospores. And again, these spores will then be windblown to be dispersed and produce more fungi. Okay, so here's showing you a picture. Again, we had this same picture in our lab manual too. We looked at the mature structure of the mushroom, and we identified some of these major parts, right? The gills, the stalk, the cap. And basically you can follow the reproductive cycle here. Again, please don't memorize this, but I could show you this picture, and you'd be able to sort of identify. You should know the names of the basidiospore, the basidium, right? Understand this. So if I, asked you to identify this or what have you, we should be able to do that. Okay, so again, within the club fungi, I mentioned these smuts and these rusts, so a little bit more about them. In particular, they usually will parasitize cereal crops, right? Like rye and wheat and things like that. And so they are a particular problem for those types of crops. And so it's something that the farmer, the industry has to deal with, because they can decimate these really important. These guys don't form basidiocarps. They just, have these spores that are super small, and there's tons of them. And so it's easy for them to be transmitted. A lot of times, though, the life cycle will require 2 different host species. So if you don't have that other alternate host, they won't be able to grow, right? So wheat rust, so one way you can control it is by getting that alternate host, right? So I said a lot of these guys will affect cereal crops like wheat. One of the ways that we're trying to control these is by breeding new resistant strains of wheat, right? Or genetically modifying a particular strain of wheat so that it's protected against these. But apparently these rusts can mutate, and so there's new forms that are formed all the time. So I guess it's sort of this ongoing kind of fight with these fungi to keep those wheat crops. Right, so this is actually showing you a corn smut, right? So a bunch of different grains they'll affect. Ew, gross. Nobody wants to eat that corn, right? And so if you look at the wheat rust microscopically, you can see that here, this sort of fungus growing on there. All right, so I told you we're going to come back to these symbiotic relationships, the mycorrhizae, and then also lichens, which we mentioned in lab, and I have some examples, and you guys look for some other examples outside. So this is a symbiotic relationship between a fungus and a cyanobacteria or a green algae, okay? And so the fungal hyphae will penetrate the photosynthetic symbiont and basically take the nutrients directly, right? So from that green algae or that cyanobacteria that can photosynthesize, the fungus will basically take the nutrients from them. So that photosynthesizer will make food, and essentially the fungus will take it. It does seem like the fungal symbiont is really the one that's benefiting from this relationship. It says it's kind of possibly mutualistic, but basically the photosynthetic symbiont is fine on its own. That fungal symbiont needs that photosynthesizer to grow. So it seems like it's the fungus that's benefiting more from the relationship. All right, so examples, right? In terms of what they look like. So there's crustose, fruticose, and foliose. And again, so crustose is probably the one that you see most often, that when we went out to look, you see this on tree bark, right? Tree bark and bare rocks. Mostly you find it around here on tree bark. So that crustose one is the form that you see most often. Fruticose is more shrub-like, and foliose is leaf-like. And the thing with these guys is they can live in areas of extreme conditions, right? And so if you talk about areas where there's not much plant life or growth, like rocky areas or whatever, these lichens essentially come in and they can start to kind of break down things, right? And then basically can contribute to soil formation. Once, then that area becomes more suitable for other growth, other plants will basically infiltrate that area. But if these guys didn't get there first, it wouldn't be possible. They're also super sensitive indicators of air pollution. So scientists, I think, environmental scientists will sort of use these guys as sort of a measure of how much air pollution is around. All right, so this is showing you this. Again, if you have this little drawing here, you see all the fungi hyphae, and this is the algae, right? A green algae or a cyanobacteria, and essentially these hyphae just reach in and basically steal their food, right? And here are the different, there's the crustose versus the fruticose versus the foliose. All right, and I told you we'd come back to talk about mycorrhizae a little bit more. Again, a mycorrhizae is a symbiotic relationship between a fungi and the roots of plants, okay? And those fungi give the plants roots a greater absorptive area, greater surface area, so they are more successful in obtaining nutrients and minerals from the soil. Okay, so it's usually a sac fungus, but we kind of these are a separate group now, right? So it's mycorrhizae, or we call them AM fungi. Remember? So this is kind of referring back to them as ones kind of being part of that group. So bottom line is, remember these are been categorized into a separate group, right? So if we go all the way back, sorry to arrow back, it's just easier that way than me going out here, right? These are AM fungi, okay? So if you're talking about fungi forming a symbiotic relationship with plant roots, you call that sort of relationship, or those fungi with the plant roots, mycorrhizae. The name of the fungi or the group is AM fungi. Okay. All right. So anyway, you can kind of have two different types in terms of, so basically the hyphae of the fungi penetrate the the plant roots. Okay. And so some of these can kind of grow between the cell walls of the plant. Others can actually penetrate the cell walls, okay? And so there's these ectomycorrhizae versus endo. Again, I'm not going to really ask you to kind of distinguish between the two or identify. You can actually look at a microscopic slide and you can identify the difference between an ectomycorrhizae versus an endo. Okay. Yeah, so actually, right, those AM fungi, like we said, are separate, a separate group that are particularly involved in these symbiotic relationships. There are other examples of other types of fungi that can form these types of relationships as well. So truffles live in association with oak and beech tree roots. And so if you're looking for truffles, right, pigs will sniff out these truffles. Obviously, you're going to look near these oak and beech trees. So anyway, there's sort of a long history of these symbiotic relationships between plant roots and fungi, and that these fungi really benefit the plant and allow them to take in many, many more nutrients than they would be able to do without. Okay, so like super simple drawing here. Here's your plant without any fungi associated with its roots. This guy has a lot. It grows much taller. Okay, so like if I showed you this, you'd be able to tell me that this plant has mycorrhizae associated with its roots. Okay, or you have this relationship. All right, and that's the end of our fungi lecture.